Our brain is the most complicated organ in our body. It is responsible for many of the actions we do in our daily activities. From wiggling your toes to typing your 10-page paper that's due at 11:59 that night. But how does our brain tell our thumb to fight in a thumb war, or tell our body to do the worm when we dance? The answer: neurons, millions and millions of neurons. So, how do the neurons tell us what to do? Well, a signal travels through the neuron to another neuron, which travels to another neuron till it reaches its destination, causing the action. But examining all the neurons involved in moving your big toe is beyond complicated, so we'll focus on two neurons for now. We start with two neurons. Let's name one the presynaptic neuron and the other neuron the postsynaptic neuron. Both these neurons are composed of a cell body, dendrites, an axon, and an axon terminal. The two neurons never touch, so there is a space between them and around them. The space around the two neurons is called the synapse, which is filled with all sorts of fluids and funny little things. The space between the two neurons is called the synaptic cleft. The presynaptic neuron is the one that is stimulated, which causes an action potential that travels down an axon into the axon terminal. In the terminal, there are many synaptic vesicles. These vesicles contain neurotransmitters inside of them. The action potential causes the calcium ion channels to open and allow calcium to enter the cell. This causes the vesicles to release their neurotransmitter. Through exocytosis, the neurotransmitter then diffuses through the synaptic cleft and binds to its receptor. The neurotransmitter can then combine to an ionotropic receptor or a metabotropic receptor. If the neurotransmitter binds to an ionotropic receptor, the neurotransmitter acts as a key and opens the ligand-gated sodium channel. If it binds to a metabotropic receptor, then the neurotransmitter releases a molecule that indirectly opens the ion channels. Once the ligand-gated sodium channels are open in the postsynaptic neuron, the impact can be excitatory or inhibitory. Under excitatory, positive sodium ions go into the cell, making the membrane potential more positive. If enough sodium ions enter and they hit the threshold at negative 55 millivolts, nearby sodium channels open and allows more sodium ions to enter. This is known as depolarization, as the depolarization continues to move, causing more sodium channels to begin to open. This moving depolarization is called an action potential. As more and more sodium ions enter, the intercellular portion of the cell becomes more positive. Once the interior reaches roughly 35 millivolts, voltage-gated potassium ion channels begin to open. Once the potassium ion channels are open, positive potassium ions flow out of the cell into the extracellular portion of the cell. This is known as repolarizing. This repolarization allows the membrane to return to its initial state, also known as its resting potential, negative on the inside and positive on the outside. This process follows the action potential as it propagates through the neuron. Now, what if the effect is inhibitory? Instead of working with sodium ions and potassium ions, chloride ions take the place of sodium ions and potassium ions stay. Similar to sodium ions, negative chloride ions move into the cell and potassium ions move out of the cell. Since the inside of the cell originally is negative, the negative chloride now in the cell will make it even more negative, causing a hyperpolarization instead of a depolarization. The hyperpolarization will make it more difficult for the membrane potential to reach threshold, making it less likely for an action potential to occur. But we still have one unanswered question. What happened to the neurotransmitters? If the neurotransmitters stayed in the synaptic cleft, then the ion channels would be continually active, which would make the membrane unstable. Luckily, there are a few ways to deactivate a neurotransmitter: degradation, reuptake, and autoreceptors. Degradation deactivates a neurotransmitter by using enzymes located in the synaptic cleft that specifically break down neurotransmitters into a substance that has no effect on the channel. Reuptake is simpler. The neurotransmitter simply re-enters the presynaptic cell through the channels in the membrane. Autoreceptors act similar to a thermostat. When there are too many neurotransmitters released into the synapse, it decreases the release of further neurotransmitters when the action potential arrives. All these cause the membranes to stay stable and allows for the neuron to remain stable as it communicates to other neurons. And that, folks, is how our brain tells us to do the hokey pokey.